Every specimen collection should be done properly and accordingly. In this scenario, a physician is filling out a test requisition form for a patient that needs a laboratory test. A test requisition form requires specific information to ensure that the right patient is tested, the physician's orders are met, and the correct tests are performed at the proper time under the required conditions. Here, a registered medical technologist reviews the test requisition form filled up by the physician to make sure that everything is accurate and in order. The registered medical technologist now collects the blood sample. After collecting the blood sample, it is important to label the specimen properly using an evacuated tube. After blood collection, proper tissue processing includes blood smear technique. Blood smears are useful for examining cell morphology and identifying blood-borne parasites. Making great blood films takes practice. We should not be discouraged by less than perfect smears. We need to submit all blood smears, perfect or otherwise, as areas of the film may be suitable for examination. Our equipment needed includes clean microscope slides, capillary tube, the evacuated tube with the extracted blood, and a pencil for labeling. Our first step is to leave a drop of blood about 4 mm in diameter on the slide. Next, hold the spreader slide or second microscope slide at a 30 to 40 degree angle at the end of the slide. Using a smooth motion, draw the spreader slide back through the entire drop of blood. Allow the blood to spread evenly along the edge of the spreader slide. Push the spreader slide forward along the length of the lower slide, maintaining a constant smooth motion, angle, and even contact.
In streaking technique for smear preparation, the specimen is rapidly and gently applied in a direct or zigzag line throughout the slide, attempting to obtain a relatively uniform distribution of the secretion. Step 1. Pour a small amount of the sediments on end of a clean glass slide. Step 2. Using an applicator stick, streak the sediments in a straight or zigzag line throughout the remaining length of the slide. If a wire loop is used instead, collect a loopful of the material and smear the material on the slide. A relatively uniform distribution of the material should be obtained. Cover it with a cover slip. Step 3. Label the slide.
The second technique is spreading. A portion of a material is gently spread into a moderately thick film by teasing the strands apart with an applicator stick or a wire loop. It is recommended for smear preparation of fresh sputum, bronchial aspirates, and thick mucoid secretions. The motion of the applicator stick comes in a longitudinal pattern or straight line. This is a simple technique similar to how butter is spread on the bread. Steps in spreading. First, transfer a small amount of the sediment on one end of a clean glass slide. Using an applicator stick, spread the sediments throughout the slide. Lastly, label the slide accordingly.
This is done by placing a drop of secretion or sediment upon one side and facing it to another clean slide. The material disperses evenly over the surface of the two slides in the opposite directions may be necessary to initiate the flow of the materials. The two slides are then pulled apart with a single uninterrupted motion and the specimen placed under the microscope for immediate examination.
Touch preparation is a special method of smear preparation whereby the surface of a freshly cut piece of tissue is brought into contact and pressed onto the surface of a clean glass slide, allowing the cells to be transferred directly to the slide for examination by phase contrast microscopy or staining for light microscopy study. In touch preparation, the cells may be examined without destroying their intercellular relationship.
Hi! We are Group 6 of BSMLS 2M and we are about to show you the procedure in conducting a gross examination, fixation, decalcification, dehydration, and clearing of a tissue specimen. Before we start, I would like to give a very brief overview about the purpose of these procedures. Fixation with the use of formalin is done to prevent the tissue from alterations that may lead to its decomposition. Since our specimen is a chicken liver, we do not need to conduct decalcification because it is only done to calcified or hard tissues, such as bone and teeth or arteries with atherosclerotic plaque formation. The purpose of decalcification is to soften hard tissues for easy sectioning in the later procedures. Next, dehydration is the excessive buffing of the specimen with increasing concentration of alcohol and the purpose of which is to remove free water from the tissue. Next, clearing is the removal of the dehydrating agent or alcohol by immersing the specimen in saline or our clearing agent. Now that we have an overview of the procedures, we are going to start with our gross examination. Gross examination is done by a pathologist and macroscopic characteristics of the tissue submitted are noted in detail. Before beginning, it is important to wear our personal protective equipment or PPE such as but not limited to a clean pair of gloves, mask, and lab gown. This is to prevent contamination to the specimen and to ensure safety of the personnel. As mentioned, our specimen is a liver obtained from a full-grown chicken. It is wedged shaped, the color is reddish-brown, and it is soft to touch. As you can see on the liver surface, there is no observed tissue damage or the like. Lastly, for its size, so as you can see, we have two lobes of the liver. So we're going to start in the right lobe. So the width of the right lobe is 45 millimeters. The length of the right lobe is 77 millimeters. Lastly, the height of the right lobe is 5.5 millimeters. The left lobe, the width is 39 millimeters. The length is 60 millimeters and the height is 3 millimeters.
The second step is to cut sections of the specimen. The recommended size is around 3 to 4 millimeters. In cutting the specimen, a pair of clean gloves, sharp knife, specimen of choice, and a clean chopping board are needed. I will perform a transverse or cross-section cut. This is to obtain a representative tissue for thorough examination.
You can see the chicken liver isn't hard to cut using the knife, while the sectioning reveals a squishy texture and the color consistency is still in dark red to red-brown mucosa with its tissue fluid.
Hello, so today I'm going to show you the step-by-step process of making disposable paper boats. So, disposable boats is used in the Histopath laboratory and it is very economical and very easy to use. So the materials needed in making paper boats are glossy paper or magazine sheets. So we need to cut this into the size of 1/16 sheet of yellow paper. So, and scissors. So after cutting the glossy paper into the size of a 1/16 yellow paper, this is how it would look like. And so let us move to the next step, which is to fold the paper into 3 parts lengthwise and 3 parts crosswise. So in folding, make sure that the folds are well defined so it will be easier for us to make the paper boat. And after folding, we now have 9 boxes. Let's count 1, 2, 3, 4, 5, 6, 7, 8, and 9. And the next step is to fold the 4 corner boxes in the edges. So when folding the 4 corner boxes, allow the perpendicular lines to meet. With this, the line created by the cross sectional fold and the line created by the longitudinal fold should meet or align. Like this. And we need to do the same in the remaining corners. So, after folding, we have created 4 flaps. 1, 2, 3, and 4. And the final step is to fold the flap outward to stabilize the box. And secure. Like this. So after securing, you can now fill this with the embedding medium and then later on you can put into the tissue section. So that's how we make our paper boats. Very easy and very economical.
So now that this specimen has been cut into sections, we are going to place our specimen or our chicken liver into the tissue cassette. But for the tissue cassette, we have the gauze as a substitute, for we have no available tissue cassette at the moment.
The specimen is then placed in a liquid fixing agent. As for this case, we will use formalin. And this will slowly penetrate the tissue, causing chemical and physical changes that will harden and preserve tissue and protect it against subsequent processing steps. So, if delayed, some cells may completely disappear, such as the epithelial cells in intestinal specimen obtained. So, we need to place the specimen in fixative as soon as possible after the blood supply has been interrupted.
Next procedure is to cover the beaker with foil and leave it for 18 to 24 hours. Well, gross sections of routine tissue should be fixed at least 8 to 12 hours to ensure at least adequate fixation. Artifact-free specimens could be produced with neutral buffered formalin fixation only if they were fixed a minimum of 30 to 40 hours and that profound artifacts were apparent after 7 hours of fixative exposure.
The removal of alcohol is done through the immersing of tissue in an ascending concentration of alcohol. For our experiment, ethyl alcohol is used as the dehydrating agent. After dehydration, the tissue is immersed with a clearing agent, which is saline as being used in our experiment.